Hello, my name is Jack Rinehart, and I will be presenting my research on the effects of Fragile X mental retardation protein on Drosophila melanogaster wing epithelial tissue. Fragile X syndrome is the most common form of inherited mental retardation in humans. Some of its symptoms include facial dysmorphia, macroorchidism, and autism spectrum disorders. It results from a mutation in the FMR1 gene. The FMR1 gene normally encodes FMRP, or Fragile X mental retardation protein, a negative translational regulator of brain development genes. In the absence of FMRP due to FMR1 mutation, FMRP target genes are overexpressed, and this results in defects in synaptic plasticity and therefore cognitive and physical defects. The actual mutation can be seen in this image. It's composed of a repeat expansion of a CGG sequence in the 5' untranslated region of the FMR1 gene. This expansion becomes hypermethylated, and the methylation extends into the promoter region of the gene, therefore preventing transcription of FMRP. The Drosophila melanogaster genome contains one highly conserved FMR1 homologue. This is compared to humans who contain several FMR1 related genes that work together to perform the same function. And for this reason, it's easier to study the effects and mechanisms of FMR1 in the fruit fly. The primary technique I used in this research is the GAF4 UAS ectopic expression system. This is a system used to express a gene of interest in a specific tissue. It starts with two genetic variants of flies. The first contains the GAL4 gene, which encodes the GAL4 transcription factor, and it is driven by a tissue-specific promoter. The promoter used in this experiment is DPP, or decapentaplegic. The other fly variant contains the target gene to be expressed, fused with the UAS, or upstream activation sequence. When the two flies are bred, the progeny will contain both the GAL4 gene driven by the tissue-specific promoter and the target gene fused with the upstream activation sequence. Then, in the tissue of interest, GAL4 will be transcribed, and the GAL4 transcription factor will bind to the UAS and activate transcription of the target gene. The target gene in this experiment was FMR1. Decapentaplegic, or DPP, is a morphogen responsible for establishing cell polarity in the Drosophila wing. It is originally expressed at the anterior-posterior axis of the wing imaginal disc. The anterior-posterior axis is this line that goes through the middle of the wing, and the wing imaginal disc is simply the precursor structure to the adult wing that develops during metamorphosis. So my hypothesis is based on a study from a previous student's research. He found that overexpression of FMRP in the Drosophila eye results in decreased differentiation of eye neuron cells in the fruit fly. So my hypothesis is that FMRP overexpression in Drosophila wings may affect epithelial tissue structure, and specifically, it may cause a shrinkage in the wing epithelium. If so, this shrinkage may be due to one of two things. First, activation of ectopic apoptosis, or cell death, or two, cell structural defects, and in particular defects in adherens junctions. If the shrinkage is the result of ectopic apoptosis, then it may be expected that FMRP can affect the expression of apoptosis protease caspase 3. If the shrinkage is due to cell structural defects, then it might be expected that FMRP is capable of affecting the expression of adherens junction protein E-cadherin. For my materials and methods, first 3 Drosophila genetic variants were obtained from the Indiana University Bloomington Stock Center. A wild type, which was used as a negative control, as well as the 2 genetic variants necessary for the GAL4 UAS system. I then crossed the experimental flies and dissected their progeny at the third instar larval stage. This is the final stage of metamorphosis when the imaginal discs are largest and easiest to isolate. So then the wing imaginal discs were isolated and treated with antibodies for immunocytochemistry. Immunocytochemistry is a method of visualizing proteins of interest in tissues using antibodies and fluorescence microscopy. Other fly progeny were allowed to grow to the adult stage, and their wings were removed and mounted on microscope slides so that the morphology of the adult wings could be compared in the experimental and control flies. For my results, first looking at the adult wings, when FMRP is overexpressed at the anterior-posterior border due to the DPP, tissue-specific promoter, the adult wings exhibit a notch at the distal tip. And you can see here in the experimental wing, there's this notch structure, whereas the control wing features a smooth edge. Also, the experimental wing features a shrunken anterior-posterior border compartment, which I will call this compartment between these two veins. You can see that it is noticeably more narrow than its counterpart here in the control wing. And this result was quantified in this graph here. So I measured the width of the anterior-posterior border compartment in both the control and experimental wings and took that as a percentage of the total width of the wing. And I found that the experimental wings, in the experimental wings, the anterior-posterior border compartment was about 10% more narrow than the compartment in the wild-type wing. And these results were statistically significant. So since there was wing epithelial tissue shrinkage, I used immunocytochemistry first to screen for caspase 3. So on the left, I have the control wing disc, and on the right, the experimental. And on the top shows FMRP expression, and the bottom shows caspase 3 expression. So in the experimental wing, you can see FMRP is expressed at the anterior-posterior border as expected. At the bottom, caspase 3 expression does not seem to correlate with FMRP expression. You might be able to see a little bit of expression in this region here, which may correlate with the expression here of FMRP, but in general, this data is not significant enough to yield conclusive results because this correlation was not consistent across samples. I also screened for E-cadherin to determine if there may be a correlation between FMRP expression and E-cadherin expression. Again, on the left is the wild-type wing disc, and on the right, the experimental. And the top features FMRP expression, while the bottom shows E-cadherin expression. Again, you can see FMRP expression at the anterior-posterior border in the experimental wing disc. But now you can also see expression of E-cadherin at the anterior-posterior border. And this expression pattern is nearly identical to the expression pattern of FMRP. And so this suggests that FMRP may be capable of modulating the expression or localization of E-cadherin. As a follow-up experiment, I looked at a different tissue-specific promoter called nubbin. Nubbin is a cell polarity gene expressed ubiquitously in the wings, unlike DPP, which is only expressed at the anterior-posterior border. The rationale for this experiment was that since localized expression of FMRP using the DPP promoter results in localized adult wing epithelial tissue shrinkage at the anterior-posterior border, then perhaps ubiquitous expression of FMRP using the nub promoter would result in widespread wing shrinkage. So I set up a similar cross to the previous one, this time using the nub tissue-specific promoter instead of DPP, and found that ubiquitous overexpression of FMRP does result in nearly complete wing shrinkage. On the left, you can see an adult wing from a nubbin fly that was removed and mounted. The actual wing is this structure down here, which is clearly very deformed and shrunken compared to the wild type. And on the right, you can see some unaltered adult flies. On the left is the control, and on the right is the experimental. In the control, you can see the fully formed wing structure, but then on the right, in the nubbin flies, there's only a very small wing shown here. And in this view, you can hardly see the wing at all. So my conclusions. First, FMRP overexpression consistently results in shrinkage of adult wing epithelial tissue. This is demonstrated both in the DPP promoter and the nub promoter. This structural defect may or may not be the result of apoptosis due to the results from the caspase-3 screen. However, FMRP is likely capable of modulating EC adherent expression or localization, and thus perhaps adherens junction formation. And this is demonstrated by the EC adherent screen. Finally, EC adherent modulation as a result of FMRP overexpression may be a more direct cause of the wing epithelial tissue shrinkage that was observed. For future research, first we could screen using other antibodies to determine whether FMRP affects the localization of other adherens junction proteins. Since it's proposed that FMRP may be necessary for proper adherens junction formation or regulation, then a correlation between FMRP and other adherens junction proteins may be expected. Also, we need to reach a mechanistic understanding of the relationship between EC adherent expression and tissue shrinkage. There are likely many intermediate steps between EC adherent expression and tissue shrinkage, and so if we can understand the mechanisms and processes behind this, information could inform potential prevention or treatment options for Fragile X syndrome. I'd like to thank my advisor, Dr. Nam, for his instruction and support during my research. I'd like to thank the Hillsdale Laureates Research Program for funding my research, the Hillsdale College Biology Department for providing me with the resources and the education to perform this experiment, and finally my friends and family for supporting me along the way. Thank you very much.